Welcome to this extracellular matrix pharmacology symposium. We really have a treat for you today. We're going to be discussing extracellular matrix, of course, and we're going to be discussing interactions with fibroblasts in that extracellular matrix environment. We have really we have Professor Dr. Linda Van Dat that we've been working with and published with in the field of iron depletion and how does that affect the extracellular matrix and the environment. I'm sure she's gonna talk a lot about that. And then we have Nick Kugerman, I hope I pronounced that correctly, really a leader in the tumor microenvironment, and they are both. They're going to be talking to you for 20 minutes. I'm going to allow a few questions after that. And then we have Nicholas, who's done a lot of research in extracellular matrix and collagens and how are they associated with outcome. And then we'll take questions in the end. So before that, before we start the session, I just have a few comments on how we are thinking. So, of course, the matrix pharmacology symposium is coming out of the extracellular matrix pharmacology conference. And last time we were in Copenhagen, Denmark. We were 450 participants from 40 countries. There were roughly 200 high-quality abstracts and 80 talks. The weather was good and the food was excellent. And we really had a good time interacting with each other, because that's all what science is. So this year we will have the extracellular matrix pharmacology meeting again. It will be in June. It will be in Copenhagen. The weather will be good again, and there will be good interactions, good food. We're expecting a little bit more than 700 participants, meaning this is by far the biggest extracellular matrix pharmacology conference in the world, focusing on how we change the matrix in multi-organ diseases, including solid tumors, and I really look forward to that. We have really an amazing program that I urge you to look at and pencil that in your calendars. So what are we going to talk about today? We're going to talk about extracellular matrix. We're going to talk about how matrix is bad for patients. We all know that. We also know in particular pancreatic cancer is a particularly fibrotic disease. We're going to see data from our presenters today on this. And we're going to talk about how different levels of fibrosis are interfering with outcome, both because fibroblasts possibly are doing something themselves, but also because there's steric hindrance. So really fibroblasts are dangerous. One way of quantifying fibroblasts is, of course, to look at collagen production. So when we have fibroblasts that are quiescent, they are not really doing anything. Then when they become activated, then they proliferate and they produce matrix, and then they secrete collagens. And for order for these collagen fibers to be put into collagen fibrils, then they need to cleave off their propeptides, and we can measure those as surrogate biomarkers for fibrogenesis and activated fibroblasts. In fact, last year, year before, we had a measurement of fibroblast activity, type 3 collagen formation. We just call that ProC3, supported by the FDA as the first serological biomarker for prognostic enrichment for solid tumors. And what does that mean? What does that mean? Well, if we look at the pre-treatment levels of these are 3 different studies with melanomas and I would say 3 leading IOs, EP, Nivo, and Pembro. The good news is that if you look at baseline and look at those that have a low level of fibroblast activity, then they survive surprisingly well. The bad news is that the ones with a high level, the 66% with a high level of ProC3, the 33% with a high level of ProC3, they are actually or fibroblast activity, they do really bad in response to these treatments. So I'm showing this one because I want to emphasize that fibroblast activities is bad for outcome. Also because I want to hopefully inspire others to do something to manipulate those fibroblasts, because we think that fibroblast activity is important for the outcome. It's important to do research in it, and I hope you've tuned in because you're interested in the matrix and how to modulate that. So fibroblast activities, we're going to be talking a lot about that. So if we depict these data in a little bit different way, so looking at these 3 different studies, then if we look at median overall survival, just cutting at the median overall survival, then if you look at the low ones, if you have low amount of fibroblast activity, then you have an up to 7-fold longer survival if you have low amount of fibroblast activity. This is not a matter of ProC3, it's a matter of that we need to look into the tumor tissue. We need to understand that matrix and how that can be affecting treatments. I think it's impressive. So 3 smaller studies, but at baseline, just one serological measurement is able to tell patients if they're going to live between 2 and 7-fold longer as compared to the low ones. I think that is super important. And then, is there causality? Well, this is not a measurement of causality, but it is saying that we had a clinical study. Then we looked into if this antifibrotic plus chemo actually had an increase or decrease in ProC3. And the patients that decreased, they live longer. I'm not sure it's because there's something causality in type 3 coded information, but it is saying if we change the fibroblast activity, then there's a possibility that patients are living longer. And with that, we're going to start the discussions from our panelists, or not discussion, but the presentations. We're going to start with Edna. I can't wait to see what you're going to share with us. Oh, I should say that I've forgotten all the technical stuff. I got excited. I'm sorry. I should ask people to ask questions in the chat, and please make some tough questions. If we do not answer them during this, we'll make sure that our presenters and also we're getting back to you. And again, thank you for joining, and thank you for our speakers for joining us this. Back to you, Edna.
Thank you so much. I hope you can hear me and see the presentation. Thank you, Morten. This is perfect introduction for what I'm going to present today. So this is just the first slide. You can scan and go to the lab website. And these are my disclosures. And here's where I tell everybody my cultural bring up, where I can tell you that collaboration with industry to me is as useful and as scary like alebrijes. All right. So what I'm going to talk today in the 20 minutes is I'm going to give an introduction to justify the idea that we need to normalize the microenvironment, the stroma itself, as opposed to getting rid of it. Then I'm going to explain what type of model we use to study cancer-associated fibroblasts and their self-generated extracellular matrix as a functional unit in vitro and in vivo. And then I'm going to tell you a little bit of how we can use this model and everything that we do preclinically to actually attempt to make a difference in patients by focusing on the microenvironment. All right, so let's start with the rationale and a little bit of introduction. So I would like to start by being big picture. Cancer is not a cancer cell genetic disease only. It actually is a full organ, and in fact, it's a systemic full body disease. So a patient undergoing cancer has an entire pathophysiological change in their entire system. The easiest way to understand this or think about this is an analogy with the ecology. And so if we think about ecological systems, then we can understand that the local niches that we're talking about that are undergoing the cancer changes have also microenvironmental very important role, because the niche could alter the function of the species that live within an ecological um, um, homeostasis. All right. The second idea, continuing with the same line of thought, is everybody knows that Paget, but actually Langhans, almost 10 years earlier, thought about metastasis as if it were a seed and soil problem. So only when there is a fertile land that the seed can germinate. And we now have beginning to, for a long time, we're thinking about the same analogy, but also about tumor initiation, not just metastatic growth. And this is very important because what this suggests is that from the get-go, the microenvironment is tumor suppressive, and something needs to change in order for the seed to germinate and get us this cancer weed. And so if that were to be the case, then we have two opportunities to target the microenvironment in order to control or prevent cancer. The initial one is never allow it to change into tumor progressing or tumor supporting and try to intercept or inhibit tumor onset. The other one is to dry the land and try to control it after it has already begun. And this is true for metastatic as well. So with all of these analogies, it is rather easy to understand that the cancer soil is the tumor microenvironment, and therefore we need to understand what types of cells reside in this soil to understand how this land is being worked to become fertile or stay unfertile. And so in epithelial solid cancers, it is very well known that the connective tissue component of the organ constitute this microenvironment, and the interstitial extracellular matrix, which Morten has spoke about, collagen, fibrous collagen-rich microenvironment, is that scaffold, biomechanical and biochemical scaffold, where all of these cells reside. And therefore, we need to understand that the fibroblastic cell, which is the local resident cell of the organ in that connective tissue, is the key cell that regulates all of this microenvironmental soil unit. And so fibroblasts are very biomechanical and biochemical, and they can change their function based on the matrix they reside within. So they not only make this matrix, but there is a vicious cycle between the matrix and the fibroblast that become a functional unit where both of them function as to sustain and help drive the homeostasis of the tissue. So in healthy circumstances, this is a healthy homeostasis. Yet during cancer, we have a new equilibrium whereby this is a different type of biology being driven by this unit. So understanding the unit is enormously important, and the easiest way to understand this unit is to think about wound healing, because it's easier for us to think about the fibroblast with its matrix as a unit if we begin thinking about a wound. So think about a wound when there is some severe, a perturbance to the organ. So under these circumstances, there is a change biochemical and a biomechanical change as well. Let's imagine that these are adjacent to where that happened. So the fibroblasts that are very, very near to this perturbance are going to start moving and sensing this change that just happened, and they are going to populate this wound. This wound can be already clotting, so there is a lot of fibrin, for example. So now the functional unit of the fibroblast together with fibrin start working very similar to what we know as the fibroblastic reticular cells in the lymph node, which is an acute type of reaction to clear the wound. Let's get rid of the bacteria or anything, or dead cells or anything that is there, clean up. And after that, and during that, the fibroblast itself starts changing this unit by getting rid of this transition matrix made of fibrin and beginning to make a more permanent matrix made of these fibrous polymers that reside within a scar, for example, or a wound healing, which is beginning of that interstitial natural matrix, which is the fibrous matrix. Under these circumstances, now the unit changes again its function, and it begins a little bit of inflammation that can be understood by fibroblasts talking with fibroblasts, for example, via the matrix. This is known as nemosis, which is an inflamed way of fibroblasts to alter the immediate microenvironment and systemic microenvironment, while at the same time beginning the wound, the proliferation and regeneration of the organ that has been severed, for example. These cells are naturally then to die, hence the name nemosis. Yet at the same time, these cells, in a non-mutually exclusive way, if the wound was very big, they will begin to contract and pull these fibers that they're making as a unit, so they can actually prepare the contraction and the closure of the wound. At the same time, these cells that are secreting inflammatory cytokines and chemokines, they're also providing some nutrition for them, for the cells that are starting to populate back for the tissue regeneration, because under these circumstances, where the wound is going to end up being scarred, there's not going to be a lot of angiogenesis and new vessels. And so the fibroblast also can provide some nutrition under those circumstances. And then if this was an acute wound healing with a resolution, all these reverts myofibroblasts apoptose or die or are moved away by some macrophages. It is not a full differentiation. They're not postmitotic, and the few ones that have become postmitotic are then senescent and also normally removed by macrophages, natural killer cells, etc. What happens during a chronic inflammatory circumstance, like cancer, is that all of this, the tolerability, the inflammation, the nemosis, and the myofibroblastic contraction is consistently upheld and chronically sustained, and there is no acellular wounding and no scar being made. And all of this is really truly known as what is cancer. And so continuing with this analogy, these types of cells can also provide tolerability, and it's the same cells. So I think that what I just told you is that we have APCAFs, ICAFs, and MICAFs in their functional sense in all of these processes of wound healing, and all of these are sustained. So while the APCAFs, for example, we know that these antigen-presenting CAFs, cancer-associated fibroblasts, are actually anti-tumor to start with, they can become tolerable and then help and sustain tumor progression and onset as well. All right, so with this analogy, the key ideas that I wanted to bring up is that while we have an acute wound, it would resolve, and this is the natural anti-tumor microenvironment that protects us from developing cancer everywhere that we have any type of perturbance. And then chronic wound healing is the adaptation of the new homeostasis of the organ undergoing tumorigenesis, whereby the microenvironment is chronically pro-tumoral. So with this analogy, it is really easy to understand that if we were to remove this natural tumor suppressive microenvironment that the stroma provides and tumor microenvironment provides, then we would also be able to get rid of the potential tumor suppression, because if we eliminate the cells that can control tumor suppression, then we will never be able to target it to the benefit, the potential benefit of the patient. And what happens during tumorigenesis, while at the very beginning, this is a tumor suppressive dry land, this barrier needs to, in a cancer cell intrinsic manner, as well as in a cancer cell extrinsic manner, down the barrier of tumor suppression so cancer can prevail. And so again, the same analogy of the seed and soil, if we want to prevent this, we need to sustain the natural tumor suppression. And if we want to control it, we need to regain it to assist with any other intervention that we're trying to do. And this is the goal of my lab. This is exactly what we're trying to do in the lab. And so this is what we try and think about every day. So we utilize the pancreatic cancer model as a means to study how to normalize stroma. And the reason for this is scientific. Obviously, this is the poster child of the tumor microenvironment. Everything that you see here in blue is actually not cancer cells, and the cancer cells are the orange cells. And so this is an exaggerated tumor mass. And despite pancreatic cancer being a very, very rare cancer, this is currently the third most deadly cancer and soon to become the second most deadly cancer. Another really interesting part of the biology and the science of pancreatic cancer is that we recently learned that we are probably all walking around as adults with precancerous lesions in our pancreata, yet we're not, most of us, going to develop pancreatic cancer, which is still a very rare disease. And the reason for this is that despite of the fact that there is already KRAS mutations in these precancerous lesions, there is a difference between the two microenvironments that can be found in a precancerous lesion of a patient or a person that died from something other than cancer, and their pancreata has precancerous lesions with actually a tumor suppressive microenvironment, while the adjacent microenvironment of a patient that actually has pancreatic cancer, if you look at their benign lesions, now that microenvironment looks completely different, much more protumorogenic. So there is an evolution in the host, in the environment of the patients that actually undergo precancerous to cancerous changes. And the third reason is both scientific and personal. In 2017, I decided to dedicate my laboratory and my research to Dr. Pat Keeley because ironically, she died from her third cancer, which was pancreatic cancer. Pat Keeley needs to be remembered as a pioneer in the extracellular matrix field because she is the one that came out with the observation of the tumor-associated collagen signatures, which are denoted here to be aligned and perpendicularly oriented to the border of the tumor. And so at this time, when Pat Keeley was developing this, we were thinking about trying to mimic this type of microenvironment in vitro. So what is this model that we study in the lab? Well, we're very lucky to be able to work in a comprehensive cancer center where the clinicians are enormously collaborative, and we get fresh tissue samples from surgery from where we can isolate fibroblasts and immediately, as soon as we can, make these ex vivo fibroblast matrix units that are functional, and the fibroblast will remember where it came from based on the type of matrix that it makes, and as a functional unit, they denote the place where they were isolated from. And this is, again, Pat Keeley's aligned matrices, and you can understand that this comes from the tumor, this comes from the adjacent, this is much more natural, maybe tumor suppressive, this is tumor promoting. This is enormously important because here we can collect conditioned media, understand what is being secreted, we are mimicking what happens in vivo, we can try to genetically or pharmacologically alter one to look like the other and function like the other. We can put cancer cells on top, we can put immune cells, we can do whatever we want with these, and still is a very, very human mimetic environment. We also, in 2005, not only demonstrated this architecture, but we demonstrated that if we were to get rid of the fibroblast and then replace new fibroblasts within these matrices, depending whether the matrix was good or bad, the naive fibroblasts will become good and bad as well. So there's a vicious cycle in this field expansion of desmoplasia. And using this model, we can also co-culture other types of cells. So we have the opportunity to study fibrillogenesis and understand whether it's bad or good, but we can also understand how the matrix affects cells that come in contact with it. And in the pancreatic cancer example, this is enormously important because the pancreatic cancer cells undergo delamination very early and are in direct contact with both these and these types of environments. Okay, so what do we do if we want to translate? We have done and made this approach in smaller multi-well approach, so we can study fibrillogenesis as well as matrix-induced responses in a medium throughput manner. And that way we can both use and assess the traits of the matrix and the function of the unit, while trying to figure out which types of interventions will normalize the unit. And this is just an example where you can see a very low throughput but enormously powerful, what we can do tumor promoting to tumor suppressive switch by altering the patient-derived units utilizing pharmacological or genetic alterations. And then we can, this simple three-color, you can see that there is an heterogeneity between these two. This could be an experimental, this could be an experimental condition, or it could be a different patient-derived unit, but you can understand the gist of this idea based on the assessment. These are enormously useful because we can study the pathways. They have an enormously good resolution, and we can do it in medium throughput as well. The medium throughput will have 4 to 5 biomarkers. The low throughput can have over 30 biomarkers. And this is enormously interesting because we can still keep it as the units, and we can differentiate fibroblastic from other types of cells like epithelial to mesenchymal cells just by utilizing this approach. So within the years, we were looking for understanding traits that could be indicative of function. We know that this is a functional unit. We know that the architecture of the matrix is indicative of the function. We know that there is cell matrix interactions and cytoskeletal changes, as I told you about before, that could be nemotic or myofibroblastic. We know that all of this can alter how cancer cells react and function, and whether it is pro or anti-cancer cell promotion. We also can use this to find drivers of these units and understand that there are targets. And we can even use conditioned media or patient blood to understand what these what these units can do systemically to the entire body or the system that we're studying in vitro. With all of these ideas, we can then move forward and initiate what we call the harmonic output of stromal traits, or a host factor analysis for each patient unit that we isolate in the lab. And this is just a couple of examples where you can see extracellular components and intracellular components. And by evaluating the levels and the organization of the matrix and all of these features that I was trying to tell you about, we can actually give a number that will tell us whether these patient unit is pro-tumorigenic or not. So with this approach, how can we then think about how to use this preclinically as an example that could then maybe later on taken to the clinic? So the way that we can do this is probably taking advantage of people and not advantage, collaborating. And because we need a preclinical village, we need to understand the unit transition, and we need to do it consistently. This is just an example that I would like to bring forward, where Dr. Meyer found in 2017 that if he were to take healthy animals and irradiate them either continuously or in pulses, the nemotic environment that I told you about that is inflammatory, pro-tumorigenic, in this case there was not even cancer, is reduced. The pulses allow the microenvironment to recuperate and have a lot less inflammation. We got so excited when we learned this from him. Morten, am I late?
No, you still have 2 minutes.
Okay.
I'm
I
just.
see. I just saw you, so I got a little nervous. All right. So we started to do this in the high throughput manner, utilizing the system that I told you about, where we can do function and traits, and we gave radiotherapy to these units. And what we did was to compare pulse low dose radiation to continuous rate radiation. And we utilized these 5 ideas where we can investigate the matrix, we can investigate the canonical TGF-beta or the actin bundling or the integrin-based or the systemic features. I'm going to let you go online and see all of this, but what we learned is that none of this is indicative of the function that we saw, and we needed to utilize all of the 5 parameters to really understand the going back to tumor suppression. And the reason for this is that there is an enormous heterogeneity between patients, and some will change the matrix alignment while others will change the integrin signaling, for example. And with this in mind, we started to think about how to move to the clinic. So obviously, I'm going to not show you how we do all of these animal models and go directly to how we do it with human samples. So we need to gate on the cells of interest so we can actually understand whether we're normalizing the stroma, yes or no. We do this by multiplex immunofluorescence with an enormously beautiful resolution subcellularly, so we can ask all the pathways. We can do this in a tissue microarray manner, and we can ask specific questions. For example, Janusz can say, how many fibroblasts are in a tissue, and are these fibroblasts good or bad, based on all of the traits that we can ask with 32 biomarkers for the host factor in vivo. And he can find different cells, and he can find different functions and their spatial distribution and all of the correlations between them. So how do we get to the clinic? Well, this idea, together with our enormously good collaborators, we came up with this clinical trial where we were trying to do pulse low dose radiation compared to the regular type of radiation in total neoadjuvant therapy, where we give both chemo and radiation to patients. And then we can take a sample before radiation and a time of surgery. This clinical trial ended their recruitment, and we have preliminary data for the controls. The controls were either treated or untreated with the standard of care total neoadjuvant. We saw that there is more fibroblast. The fibroblast heterogeneity is downregulated, but what happens is that utilizing this approach, we were able to see that the more treatment, the less cancer cells, but the more dangerous the microenvironment becomes. And this is really very interesting, because these patients need to wait 4 to 8 weeks until surgery, and it could explain why in some cases, but not all, these patients will immediately recur. So we can measure whether the stroma is good or bad. And what we have been able to see is that using the continuous rate radiation, we have more basal aggressiveness and pro-cancer immunity. We have a higher host factor and less functional heterogeneity. We hope that with the PLDR, which we are now testing, we will have a lower host factor to the potential benefit of patients. So far, patients are doing wonderful. And so we want to hope that this is because of the microenvironment. But we know now that really we are causing dangers to these patients because we're aggravating their microenvironment. And so we came up with another clinical trial that also just ended accrual, and we will soon also know whether by adding this over-the-counter stroma normalizing agents, we could then relax the microenvironment before the surgery. And with this, I just want you to remember that it is worth to study fibroblasts and matrices as functional units, that the unit-guided approaches can help us to get to clinical translation, and that teamwork is dream work. And this is the village. The people with the asterisks are the current lab. Janusz is now the manager of the spatial proteomics facility. These are our collaborators, funding, and a lot of facilities that we utilize. I would like to just give a call to people to try and utilize this wonderful new journal from the AACR, open access, as the mandate in the government, at least in the United States, now calls. And I would like to thank you all, and come and visit beautiful Philadelphia. This is the lab, and this is the microscope room. And I'll take any questions if there's still time.
Thank you so much, Edna. That was really a beautiful talk and a lot of information. There are some good questions. I'm going to combine one of the questions with a super interest of my own before we move on. So, does your model allow you to look at extracellular matrix modifying enzymes such as transglutaminases and LOX? That's a question from Adriana. And then my question on top of that is, when you have done all this, I noticed that you have been looking at fibrillar collagens, type 1 and type 3, but which of the non-fibrillar collagens or which collagen is actually highly expressed? Let's start with Adriana on ECM modifying enzymes.
So the answer to the first question is yes, of course. We can do Western blots, we can utilize immunofluorescence, we can do transcriptomics, we can really figure out whether these are expressed or localized, stored in the matrices. We really, truly can do that. That is not an emphasis of my lab, but this model allows to do that. As for the which matrix proteins are there, I'm really happy that you asked this question. We have from the same patient the good and the bad units, and also pharmacological and genetic modify units to become bad or no longer tumor promoting. And what we did was to isolate those matrices first, demonstrate their pro or anti-tumor capabilities, and then digest everything based on Pat Killis and Alejandro Nava's means to digest matrices and send it to our collaborators for proteomics. And so the interesting thing is that at least for this cohort, we didn't see anything new matrix-wise when we normalize it. We only lost some proteins. Some of the proteins that we lost in the matrix were obviously all of them are matrisome-based. We found actually not, still talking about polymerized collagens, we found collagen 6 to be enormously important, and all 3 subunits of it were downregulated in just 9 different proteins that we found. So this wasn't a let's discover what proteins are there, but which proteins change when we normalize the matrix. Yet our collaborator in South Carolina, she is actually doing spatial matrix proteomics and also systemic peptidomics of matrices, and she probably will help us figure this out. We're doing this collaboration to know exactly which collagens are changing in this specific system and whether those correlate with patient systemically.
Thank you, Edna. I mean, I love your energy, and so does all the people online. The questions are flying in, and we'll take some of them in the end. Thank you so much. Our next speaker is Linda Van Dat. That's going to, there you are.
All right.
And again, thank you for joining. I've been looking forward to see some of your science. And talking to us about iron depletion, copper depletion, and some clinical aspects.
Copper,
okay. Yeah. Or are you? Yes, it works perfectly. Okay. The floor is all yours, Linda.
Okay, so I'm very excited to be here today, and I'm very excited as I hear about normalizing the collagen microenvironment, hearing about how important ProC3 is. I'm going to really offer you an opportunity to achieve both modulating ProC3 and normalizing the collagen microenvironment through showing you some of the work that we've done on copper depletion. And I hope that my team and I have been working on looking to tackle one of the toughest areas of breast cancer, which is how to prevent metastasis. And I hope by the end of this, as I show you just a part of what we're doing, that you'll be as excited as we are as to next steps. These are my disclosures. So today what I thought what we would do is that, because I know there's probably no breast cancer doctors on this call, that I would just do a little quick update on breast cancer, give you a big picture overview of our project, which is looking at copper depletion as a therapeutic strategy to prevent metastasis in triple-negative breast cancer. I decided to focus in on the effects that we see with copper depletion on the collagen microenvironment, and then show you some of the works in progress and next steps. So first, just to start about breast cancer. Breast cancer is the most common cancer in women, over 2 million cases a year, with close to 700,000 deaths per year. We have many options available, which are good options, and for that reason, mortality is decreasing across most, although not all, of the globe. And in breast cancer, when we start to think about how we choose therapeutics, we think about it with risk stratification, specifically molecular subtyping. So within that molecular subtyping universe, there's a particular type of breast cancer called triple negative breast cancer. We call it triple negative because it's negative for estrogen receptor and progesterone receptor, which are hormone receptors. And also there's no HER2 new amplification. I always say HER2 is a growth accelerator present on some breast cancer cells. While it only represents 17 to 20% of all breast cancers, it represents 50% of those who have metastasis. So it's really a problem. And as many of you are probably aware, metastases are responsible for the majority of deaths from breast cancer. So we've already heard a little bit about Dr. Paget. I never heard of Dr. Langen's ED, so thank you. I'm going to go see if I can find that paper. It was 10 years before Dr. Paget's paper, but basically he was among the first to suggest that metastasis were non-random events, gave rise to the seed and soil hypothesis. So the environment that tumors, which are the seed go to, have to be right in order for metastasis to occur. And when we think about how we've traditionally treated cancer, until the advent of immunotherapy, we used to think about treating the primary tumor and treating the metastasis. But I would say, and our group thought, well, what if we were to pull out the infrastructure the tumors need to spread? Could we prevent tumors from spreading? And the analogy for that was sort of like that you have a car that has no gas. So it's there, it's not causing any trouble, it's just there, but it can't move. Would that be tantamount to a cure? And so we started to develop a project in which we thought that we could target the tumor microenvironment. And I won't go through all of the reasons. I'll talk about them a little later while we thought about metals, in particular copper, if we could prevent metastasis. And since I'm a clinical researcher, when clinical researchers want to study something, what they do is they design a clinical trial in order to test their hypothesis. And so we designed a clinical trial in which we enrolled patients who are high risk for breast cancer recurrence in our clinical trial with an oral copper depletion compound. We embedded a lot of science into the trial so that we could really understand what is the right treatment for the right patient at the right time. And then in parallel, we partnered with the Metal Lab at Cornell. I was on faculty at the time there, looking at in vitro and in vivo. And then we initiated a bench to bedside and back again strategy. So we would observe something in the clinic, we'd go talk to the lab would do their stuff, they'd find something, we would go back to our research specimens and to see if we could recapitulate what was seen in the lab. And sort of where does this all take us? It takes us to a randomized study of phase 2 in triple negative breast cancer that is currently ongoing right now. So today what I'm going to show you is, we have so much data, but we're really going to try to focus in on the collagen biomarker piece of this. So you're probably wondering why copper, and copper is really important. It's important for many normal biologic processes, but it's also very important for processes which are associated with the process of metastasis. So that it means that it's important for such as cofactors of angiogenesis, so for beta FGF, VEGF, NF-kappa beta. It affects both RAS-MAP kinase signaling. It has been demonstrated to be important for invasion of triple negative breast cancer cells in particular. So what has been demonstrated from many groups is that they see translocation of copper from nucleus to extending filopodia. That's how cells move. Copper is important for oncogenic BRAF V600 signaling. It regulates expression of PD-L1. And actually, when you look at the hallmarks of cancer, which is in the upper corner of the slide, all of those processes are affected in some way by copper. And as you know, I've mentioned that we're interested into preventing metastatic progression as opposed to cancer initiation per se. And we know that copper is a key component of the steps involved in metastatic progression, specifically motility invasion, modulating the microenvironment, plasticity, and colonization. So there are a lot of reasons I think that copper can play a role. So specifically looking at the link between copper and breast cancer, CTR1 is a major transporter of copper into the cell. And when you look at metabric data, the CTR LC3-1A1, you can see higher levels, worse outcome. When you look at tumor stage in breast cancer, you can see higher tumor stage, higher copper. And when you look at some of the more aggressive molecular subtypes of breast cancer, specifically triple negative breast cancer and HER2 positive breast cancer, you see increasing CTR1. There are many ways to copper deplete. Usually there's an oral copper chelator that is used. It's just a pill people take. There are 2 FDA-approved drugs in the US, penicillamine and trientine. We actually, in our studies, have used tetrathione molybdate. And the way that works is that it basically inactivates copper chaperones and decreases the incorporation of copper into metalloenzymes. Wilson's disease is a copper storage disease, and there's a completed phase 2 trial of choline tetrathione molybdate, which has not been reported yet. And when, and I'm going to say it was the University of Michigan who really first started exploring how copper depletion could prevent metastasis in preclinicals and then in patients. And pretty much any model that they use shows tumor regression, specifically looking at microvessel density and decreased angiogenesis. And that led to a lot of enthusiasm to complete phase 1 and phase 2 trials in patients who had overt disease. Now, in the drug development world, if you have a drug that only shows disease stabilization, it's considered a failure. And so in the 4 phase 2 trials that they did across a variety of solid tumors, the best response they got was stable disease in about 30% of patients. And so development of that compound stopped. But there was this study that I thought made me think that perhaps they were developing it the wrong way. And this was a study in which this is a genetic mouse model which develops lung metastasis by day 205. And so, of course, they gavaged the mice with either TM or water control. On day 205, they developed their lung mets. And the TM mice did not develop lung mets. But what was interesting was when they withdrew TM therapy, on average, they developed lung metastasis at a median of 2 weeks later. So this just suggested that those metastases were there just trying to sort of get a hold and grow, but for something was stopping it. And so this made me think of my breast cancer patients who can have dormant and occult disease for years and years and years. And so I thought that we could, that was where the value of copper depletion was in this setting, perhaps pulling out the infrastructure the tumors need to spread. And because I'm a clinical trialist, I designed a clinical trial in order to test this hypothesis. So in our trial, so in 2007, we launched a pilot trial of tetrathione molybdate in high-risk recurrent breast cancer. The most important thing was the patients had to have no evidence of disease, which is what NED stands for. We enrolled 75 patients. We gave patients oral daily TM for 2 years, and we picked 2 years just based on that was the longest time that anyone had been on a biologic at the time we designed the trial. And our goal was to copper deplete patients to about 30% of normal. So the way that we measured that is that we looked at something called a ceruloplasmin, or CP, and the lower limits of normal is 17. So we wanted to sort of have it between 10 and 17. The primary endpoint of our trial was looking at something called bone marrow-derived progenitor cells. These are VEGFR2 positive EPC. And we had done a lot of work in this field because VEGFR2 EPCs have been associated with tumor progression in both preclinical as well as in breast cancer. That was our primary endpoint. Our secondary endpoints were progression-free survival, overall survival, and then we had there was a lot of science built in. I'm going to show you in a second. So in terms of how do we define risk, our risk was our stage 3 and our stage 4 NED breast cancer. A stage 4 NED breast cancer are basically patients who had their cancer come back. Somehow they got rid of it. We didn't care how. It could have been radiation, surgery, chemo. But when we used every modality we had available to us, we did not find any cancer. These patients 100% recur. About 2 years into the study, it became apparent as the science advanced that a stage 2 triple negative breast cancer had the same risk of recurrence as a stage 3 non-triple negative breast cancer. So we expanded our enrollment and to allow those to be entered. As I mentioned, because we thought of this as an anti-metastatic therapy, our patients could not have metastasis. And the way that we sort of ascertained that at the time was physical exam, blood tests, including tumor markers and imaging. Patients had completed what was considered standard treatment, and all these patients were looking for something extra to try to make sure that, trying to do everything they could to try to prevent their tumors from coming back. But they'd all completed what was considered standard treatment. I always say these ladies were incredibly dedicated. They came to see us once every 4 weeks. At the time, we examined them, we did bloods, we checked their CP levels. In real time, we did flow cytometry looking at bone marrow-derived progenitor cells, and we evaluated them every 6 months with imaging to make sure they were still NED. We completed our accrual in 2014. So just to make sure that it's clear, our goal was to copper deplete patients to the point where normal cell function could take place, but copper-dependent processes could not. The way I always think about it is that there's a copper pool, and for some reason, there's cells lined up to drink from that copper pool. And for some reason, normal cells get to drink first, cancer cells are at the back end, and by the time they get to come up to that pool to drink from the copper pool, there's nothing left. So we enrolled 75 patients. We had 51 patients complete 2 years of TM therapy. 24 patients came off, 12 for recurrent disease. At the end of 2 years, our second patient who was enrolled we thought it was pretty unusual that she hadn't recurred. So we thought, well, gee, maybe we're doing something. So we designed a series of extension studies in 2-year increments to study prolonged copper depletion. And we rolled patients over on those we rolled patients over onto 2-year increments. We were, I'm going to call it a victim of COVID too, because in March of 2020, our drug supplier on which our IND was based decided that they weren't going to make our drug anymore. So we transitioned patients to TM study. And just to sort of give you a sense as to the types of patients we enrolled in our trial, 60% had stage 2 or stage 3 breast cancer, 40% were stage 4 NED. When they were adjuvants, on average, they had 6 positive lymph nodes. And when they had stage 4 disease, you can see the different sites of disease that they had. 48% of the patients were triple negative breast cancer. So just to sort of give you some information on this, so we were able to copper deplete patient using ceruloplasmin, and that was sustained over time. I didn't really talk so much about our bone marrow-derived progenitor cells, but they decreased, which is good because this is an important step in terms of relapse. And just to sort of, when we look at the time course of bone marrow-derived progenitor cells and relapse, basically what we see is that after there's a spike in the VEGFR2 positive EPCs is when you have a clinical relapse. So these were decreased with decreasing ceruloplasmin. So that was our primary endpoint, which we hit. Copper depletion was safe and well tolerated. When we looked at adverse events, the worst one we saw was grade 3 neutropenia in 1.7% of patients, and it's reversible. You hold the TM, the counts pop right back up. And that was in the setting of when we saw that if patients got a PPI, we could improve absorption of the TM. And so we gave everybody a PPI, and we made most people neutropenic. So learning experience right there. So these are the overall event-free and overall survivals out of the trial. What you can see is that there's a little bit of a drop-off in the beginning, and then the curves flatten out. I will tell you, and our median follow-up is 10.4 years. I will tell you I am in contact with all these patients, and they're all fine, although we can't follow them technically on study. But what really caught our attention was our data in triple-negative breast cancer. So if you see here, this red line here, this is the event-free survival for our stage 4 NED patients that have triple-negative breast cancer. And I'm going to say comparable data at this time, median survival for stage 4 is 11 months. So this really made us think that there's something special about triple-negative breast cancer and copper depletion. And on the right-hand part of the slide is just some of the things that we've looked at scientifically and along with biomarker development. I'm not going to really go into them. So as I mentioned, we, in parallel, have an amazing relationship with the Vivek Lab at Cornell, and Divya Ramchandani was the postdoc at the time. I was a faculty at Cornell at the time. And so we went back into the lab, and our goal was to sort of figure out, are we affecting any other process which promote the process of metastasis? And so in order to do this, we looked at triple-negative cell lines stably expressing luciferase in transgene SCID mice. Half of them got water control, half of them were TM treated. And then we evaluated the primary tumor size as well as metastasis. And so first thing, of course, is that we're able to copper deplete the mice using CP oxidase. Surprisingly, we saw no change in primary tumor size by BLI. But what we saw is there was a marked reduction in metastasis, as you can see here. When we looked at the pre-metastatic lungs, and this is where it really gets interesting, is that we saw that LOX, lysyl oxidase, with the blue bar, it's markedly reduced in the TM-treated TM-treated lungs. What that does is that reduces collagen cross-linking and reduces collagen fibrillar length, as you can see. So with that, we said, okay, well, that's really interesting. Let's go see if we can recapitulate what you've seen in the lab in our patients with breast cancer, because LOX is very, as you all know, LOX is very important in terms of creating the pre-metastatic niche in the process of metastasis. And so while we went back and it was easy, we did ELISAs on our banked specimens, and we saw that lysyl oxidase was markedly reduced in our patients who were copper depleted. And we also saw that outcome was better in our patients who had no evidence of disease, who had lower copper levels and lower LOX levels as compared to those who had progression of disease. So the next thing was, well, how are we going to measure collagen biomarkers in patients? We were sort of thinking about this because we wanted, again, we were excited when we saw that LOX levels were decreased, but, we didn't have a tumor that we could really look at in order to look at, in patients who were on study. And so, so this is where it's karma. So it was ASCO annual meeting, either 2015 or 2016. ASCO is the American Society of Clinical Oncology annual meeting. Usually there are about 40,000 people there. This is a picture of a poster session. And I was going up and down the posters when I met Cecilia Badger from Nordic Biosciences, who was presenting a poster on collagen biomarkers I don't even remember what disease type. She then introduced me to Nicholas. I then was able to sort of see the work that was coming out at about the time that we met, and we were able to embark on a collaboration in order to measure collagen biomarkers in patients. So this was the panel that they had at the time in order to begin to evaluate patients. And they told me how much specimen they needed. And I was like, oh, that's too much. So let's see if we can narrow this down a little bit more. And so what we did first is we looked at an exploratory cohort to identify relevant collagen biomarkers in 132 patients who had anywhere from stage 1 to stage 4 breast cancer. At the end of the day, we had 4 candidate biomarkers that bubbled up to the top as being significantly different between healthy and cancer. That was specifically LOXL2, collagen C1m, C6m, and ProC3, which you've already heard from Morten. So then what we did is we applied that cohort, those 4 markers to our TM cohort. And this is what we found. So looking at LOXL2, so red is no evidence of disease, and blue is progressive disease. So what you can see is over time, we did baseline 5 months, 11 months, and 24 months. Basically, what we saw with LOXL2 is that patients with lower LOXL2 were more likely to be alive as compared to those who had higher levels tended to relapse. When we looked at collagen type 1, C1m, what you can see is that the higher C1m was, the more likely to be NED, lower C6m, and lower ProC3. So the bottom line is, as you've already heard a little bit from Morten, is that there was an improved outcome associated with more degradation of collagen than formation in patients enrolled in the trial. And what we saw also was that there was an association of collagen type 1, C1m, and ceruloplasmin by disease status. So lower the CP level, and higher the C1m, more degradation. And so what we thought, and you've sort of heard this already a little bit, that at least with copper depletion, we believe that we were normalizing the collagen microenvironment, having more degradation than more dysplasia. So then, and so, why do we care? I think this group knows why we care, but just to bring it forward, we know that there's increased stiffness of the tumor microenvironment during cancer progression. And what this does is this leads to resistance of both chemotherapy and radiation. Also, at about the time, and this is a very early paper, there are many more papers, is that, we really started to look at that density and orientation of stromal ECM played key roles in migration of immune cells. So beginning to think about how, how are we going to sort of soften up that ECM so that the therapeutics that we're using can get to its targets. And so in order to do this, we went back to the bench. And then we started to look at the effect of copper depletion in an immunocompetent model. When we looked at flow cytometry in the primary tumor, we started to see that we saw a reduction in myeloid-derived suppressor cells in the TM-treated lungs. We also saw an improvement, an increase in CD4 T cell infiltration in the TM-treated lungs. And we also saw a decreased collagen density in the TM-treated lungs. So we're starting to think that copper depletion can begin to improve immune cell infiltration by reducing collagen density in the pre-metastatic lungs. We also saw some other changes in the immune microenvironment. This is work that's actually ongoing. Basically, we saw an increase in PD-L1 expression. We saw an increase in antigen-presenting cells. And we've already talked a little bit about the reduction in myeloid-derived suppressor cells. So that's sort of where we are there. I will tell you that we just got back the data on the cytokines, TGF-beta, from our clinical cohort, and that's currently being evaluated. So in terms of, we did a lot more work, and so it always comes down to how does copper depletion prevent metastasis, because that's really going to help us figure out what are the patients who can most benefit from this particular treatment. And just a couple of things. I don't really have time to show you all of the data, but the bottom line is while we were really focusing on the tumor microenvironment and how we remodel that, it actually turns out that the highly metastatic leading edge of tumor cells, the stem cell component, have very high copper levels, have high copper levels. They are very sensitive to TM therapy. And what we find is that you get much less disseminated tumor cells. So it's not just targeting the microenvironment, but maybe there's some component of the tumor itself. We've demonstrated that TM-mediated inactivation of complex 4 is a primary metabolic defect, which downstream affects AMP kinase. So there's a lot of work that's been done. In terms of ongoing scientific initiatives, as I mentioned, I have banked specimens, and I actually have primary tumors in patients enrolled on our original study. We're looking to really understand the effect of TM on both the immune and metabolic microenvironment with our bench to bedside and back again strategy. We've been trying to do some ctDNA in patients treated for a while. We thought we had it nailed down with Foresight, but they just got acquired by Natera, so who knows? And we're also expanding this. So, we heard about the effect of modulating the microenvironment in pancreatic cancer, and I think pancreatic cancer would be a great disease in order to study this. We have projects moving forward in non-small cell lung cancer, and we're working with some imagers now also. So in terms of, what's our next step? And that is we have a trial in triple negative breast cancer. A lot of reasons to think why triple negative breast cancer would be a good substrate, not only from our own work, but also when you look at established TNB resistance pathways, there are lysyl oxidase, there's metabolic pathways like enrichment of OXPHOS, and there's a number of reasons why copper would be a rational strategy. And so this is our trial. This is a trial which is currently open and accruing across 11 sites in the US. Patients who have high-risk recurrent triple-negative breast cancer, we call it RCB, 2 or 3 or node positive. These people have a 25 to 75% risk of tumor recurrence after standard chemoimmunotherapy. They are randomized to standard treatment versus standard treatment plus tetrathione TM. Sample size is 186, and there are a number of exploratory aims as well as secondary endpoints. Important within the exploratory aims are the fact that we're going to really have an opportunity to really understand the effect of copper depletion on collagen remodeling and metastasis. So we're really excited. And as Eddie say, it takes a village, and here I feel like it's taken a whole city. But these are the many partners in our projects as well as our funders, and we look forward to just getting the trial accrued to really understand can copper depletion prevent metastasis. So thank you for your attention. Oops.
Thank you so much, Linda. That was a great talk. I really appreciate that. There are really tons of questions in the chat, and we'll take them afterwards. Of course, I understand Loxel, because of I think you need one copper ion in the active site actually to do that. So what would be the second usual suspect an enzyme that would be dependent on copper in the local tumor microenvironment that you think could also contribute? I think it looks very interesting with Loxel. Are there other things from that angle?
Well, we're looking at a number of copper-dependent enzymes.
Yeah.
But I don't have a panel to tell you exactly what it is. One of the things I think is that one of the reasons why I think that this might be a good strategy to prevent metastasis is because when resistance develops in one area, you have like the 100 other areas that are being affected, so it doesn't. So it's not like with some of the traditional drugs we use. Copper is important for like NF-kappa beta, MAP kinase signaling. It's ubiquitous.
It's important for many things. Completely agree. That's why it's interesting. We are running out of time, and I'm so thank you very much, Linda. We'll get back to you with questions afterwards. Nicholas, I can't wait to see what you have in store for us. Hopefully, something that we're going to be impressed about, and how fibroblasts contributing to patient survival. There we go, Nicholas. You're on. Nicholas, you're muted. Okay, good.
Setting up, thanks. Yeah, hello everyone. So the title of my talk is related to clinical research. So how can we use extracellular matrix collagen fragments as biomarkers to get insights into the tumor microenvironment, to fibroblast activities and immune cell activities? And briefly recapping here, the extracellular matrix composition, the quality is completely altered in solid tumors, right? And we have fibroblasts that are overactive, they produce a lot of new matrix, and we have proteases that are degrading it, completely changing how the matrix looks in tumors, but also generating small peptide fragments that are released into the circulation, which is what we use as biomarkers, PRO-C3 and others. So cancer is a very active disease, so there's a lot of collagen remodeling going on. This was also what Morten showed you. I just want to briefly recap just what one collagen biomarker can do at baseline. Really, this is 3 cases with metastatic melanoma. So we have EP, Nivo, and Pembro treated patients at baseline. If patients have high PRO-C3 levels, they are basically not benefiting from immunotherapies, but if they have low levels, they are doing fairly good. So what is PRO-C3? Well, Morten also already told you. Again, immature collagen, fibrillar collagens have propeptide. They are cleaved off, they end up in circulation. We call that PRO-C3. They are a surrogate of cancer-associated fibroblast or fibroblast activation, producing collagens that are put into these fibers in the tissue. And as also mentioned, we have a letter of support from FDA encouraging PRO-C3 as a tool for clinical trial enrichment and for stratification of patients with solid tumors, basically for looking into tumor fibrosis as a component to the cancer biology. Here's a cellular link between fibroblast and PRO-C3. So when we take out fibroblast from a benign human donor from the pancreas, we can see that the PRO-C3 levels are fairly low compared to a donor from a tumor here where cancer-associated fibroblasts are derived, and they are much more active. We can play around with TGF-beta, so inducing with TGF-beta or blocking with TGF-beta inhibitors, and then we can change the levels of PRO-C3. The interesting part here is, of course, that PRO-C3 levels are directly correlated to how much net collagen deposition, how much zero red staining we eventually end up with. So the more or less PRO-C3 you have being derived from your cell cultures, the more or less collagen you will have deposited in the end. So it's a surrogate of fibrogenesis. Recently, we have been looking into different cancer-associated fibroblast donors because what is actually the difference between, are there differences between the activities of cancer-associated fibroblasts, or are they all the same? So we have here 4 human donors that provided us fibroblasts, cancer-associated fibroblasts, and they were all characterized by the same CAF markers as we know them here. But interestingly enough, when we look at the PRO-C3 levels here, we can see that there are very steep, significant differences. So we have 2 fibroblasts here that are intrinsically very active and produce a lot of type 3 collagen, meaning that they are very fibrotically active. We have 2 other donors here where they are much less active. So even though fibroblasts here are characterized by the same surface markers, their fibrotic activity can vary quite a lot. And that is, of course, important, taking into account what I'll show you with metastatic melanoma. But that actually goes from many different solid tumor types, if not all solid tumor types, in the sense that patients that present with very high levels of PRO-C3 at baseline, they have very poor outcome. So we have pancreas cancer here. You can see the scatter plot. Most patients are highly above the reference range. If patients present with high PRO-C3 levels at baseline, they do worse than if they have low levels. Likewise, in hepatocellular carcinoma, from the CheckMate 040 trial, patients treated with NEMO, high levels of PRO-C3 as baseline gives us much worse outcome than if patients present with low levels. Similarly, for colorectal cancer, this is bevacizumab treated patients, high levels, much worse outcome than low levels. Then I want to double click on pancreas cancer here, where I have an example of, is this a unique biology, or how does this really compare with other risk factors that we typically assess in cancer? So for that, we did a multivariate analysis and included, together with PRO-C3, other risk factors such as age, metastatic burden, you can say, stage of disease, CA19-9, and performance status. And even when including all these pretty strong risk factors for outcome, PRO-C3 remains an independent prognostic signature for poor overall survival, meaning that it is pretty strong but also unique risk factor for outcome in patients with pancreas cancer in this case. Another example is from cholangiocarcinoma, biliary tract cancer, similar pattern for survival outcome. Patients with high levels do worse than those with low levels. But here I also wanted to show you a scatter plot where we reflect on, first of all, patients with benign disease compared to the different stages of disease that you can have here, classified as either resectable disease, locally advanced disease, and metastatic disease. And throughout these different stages of disease, we do see patients with very high levels of biomarkers. So again, the fibrotic activity is somewhat independent of the stage of the disease. Finally, I wanted to show you breast cancer, coming from what Linda showed you in triple negative. We have here metastatic breast cancer, where we looked at PRO-C3 at baseline. So patients were treated with trastuzumab, and if levels were high in terms of PRO-C3 at baseline, median overall survival time was modest, 5-fold less than if PRO-C3 was low. So all in all, I've shown you all these different solid tumor types, some of which supported the letter of support that we were given by FDA, and they all showed the same thing, that if you have a lot of fibrotic activity at baseline, you have worse outcome. So that's all single-arm studies, and we cannot really say much about whether it's prognosis and prediction, or definitely prognosis, but what about prediction? We have a handful of examples now published where we also look into whether PRO-C3 can be used for predicting response to given treatments. One example here I have included today is from ASCO last year, presented by David Brown. It's BMS data from the CheckMate 9 trial, so that is renal cell carcinoma, and patients were treated with neocarbo or with sunitinib. So immunotherapy in combination with cabozantinib or tyrosine kinase inhibitor alone here. Patients were stratified into whether they have high, medium, or low levels of ProC3 at baseline. And you can clearly see that if ProC3 was high on nivo-carbo, patients survived just as well as they did on the tyrosine kinase inhibitor here. But if ProC3 was low, patients were benefiting much better from the nivo-carbo within arm, but also across comparing to the TKI. So this is just one example of how we can also, of course, depending on the mode of action, predict response to treatment as well. And what about bringing down some of this fibrotic burden, bringing down some of the levels of ProC3 on treatment? Is that also beneficial for patients? And clearly it is. So in a recent study that we just presented at ASCO GI in refractory metastatic pancreas cancer, patients that were treated with the double nivo-ipi immunotherapy on top of radiotherapy, there was still a few patients, a fraction of patients that were decreasing in ProC3, and they did substantially better than if they were not decreasing in ProC3. So what I'm saying with this slide is, can we somehow reduce the fibrotic activity in these patients? That will also manifest on a better and longer survival outcome. Here we have the threefold difference in 6 months overall survival time. So it is actually substantial even in this hard to treat patients. So in summary, for ProC3, it's almost what we here call a unicorn biomarker. Biologically, it's produced by calves. It's a surrogate for collagen deposition. It's elevated across disease stages. It's elevated in multiple indications. It's prognostic for outcome. We have the letter of support, independent of many other risk factors. It's associated both with response rate, but also with prediction of response at baseline. We can somehow figure out what treatment to give patient actually. And if we're able to decrease ProC3, and we should have more treatments trying to do that, then that's actually also correlate with better outcome for patients. ProC3 is of course only part of the tumor microenvironment, part of the fibrotic component, and to track the whole processes, we need a little bit more biology, a little bit more biomarkers, and we have a panel of biomarkers that we can use to assess fibrotic activities. The common denominator at baseline is that if patients have high levels of these collagen fragments related to fibrosis, they do worse in terms of outcome. Another aspect is also when we track patients over time, and this is also in the line of what Linda was showing you, showing here pancreas cancer patients tracked for 12 weeks in a suboptimal dosing in a phase 1 study, so we were not expecting any responders, but there were 6 patients that maintained with stable disease, and they also have stable biomarker levels, whereas the 3 patients that happened to progress already early on, they very rapidly increased in the collagen remodeling turnover here. So stable disease, very stable collagen turnover, but as soon as patients progress, that also manifests on the disease activity and the collagen turnover. And then we can measure not just type 3 collagen, or not just ProC3 for formation of type 3 collagen, but we can also measure degradation fragments being released out of the mature collagen. And one of those is fibroblast activating protein, which is of course also related to fibroblast activities. So I wanted to bring that in as well. This is a fragment that is specifically generated when Fab, which is a membrane protease on fibroblasts, cleave type 3 collagen, and we have a biomarker for that. And we know it's very specific for that particular epitope of interest. So we have fibroblast activating protein biomarker, that is one aspect of fibroblast biology, and we have ProC3, that's another aspect of fibroblast biology, formation and degradation, if you like. We recently conducted a collaboration with Herder Gentofte Hospital, a study on pancreas cancer patients where we looked at ProC3 and C3F, as we call this Fab biomarker. We looked at baseline, we could see again ProC3 prognostic for poor outcome with high, the same for C3F. But interestingly, these 2 biomarkers are not at all correlated, suggesting that they reflect different biology, but also that they may be complementary. So when we combine these two out and looking now only at those with low fibrosis, low PRO-C3 and C3F, we get an even better segregation between those that are benefiting the most from treatment. And you also see the hazard ratio here adjusted, 65% reduced risk of dying actually. And you see the objective response rate threefold different between low and high fibrotic activity at baseline. So we can do a little bit more than PRO-C3 and combine fibrotic signatures. Another aspect that we did some time ago now with published together with Thomas Cox and Raghu Kaluri, where we looked at what is actually the collagen expression pattern in cancer-associated fibroblasts compared to benign fibroblasts, and we used human tissues from pancreas. Interestingly, what we found here was collagen 5, collagen 8, and collagen 11 and 12 is only found in tumors and not in benign tissue. So probably there's a window here to look for more tumor-specific collagens. We dived deeper into this and mapped out the different fibroblast subtypes that you have, and it was clear that collagen 11, collagen 12, 8, and 5 was exclusively produced by cancer-associated, myofibroblastic cancer-associated fibroblasts. So we have here biomarkers that we can potentially use to reflect that biology, and that's important because this is really the fibroblasts that you want to target, that you want to get rid of in order to normalize the microenvironment in the tumors, as we've been talking about earlier today. So focusing on type 11 collagen is an example here. We have a case control study just to show you how elevated type 11 collagen synthesis measured with PRO-C11 is across indications. The area on the curve from healthy individuals is 1 or 0.99. We have the cellular link here. If we take out cancer-associated fibroblasts and culture them in vitro, we get out propeptide from type 11 collagen. We do not see any signal in normal fibroblasts. Prognostically, we have a lot of data. Again, patients with a lot of PRO-C11 in their circulation at baseline, they have worse outcomes compared to low levels, again, supporting that MyCAF activity is bad, as we would expect. And with treatment bringing down the burden of MyCAFs or reducing PRO-C11, what we also see here in pancreas cancer, that in the patients that have a reduction in PRO-C11, they do much better. At least there are threefold more patients still alive after 6 months if PRO-C11 goes down compared to if not. So PRO-C11 is one MyCAF biomarker. We have also tools to assess type 5, type 8, type 12 collagen. They are all highly elevated and prognostic for outcome in solid tumors. So what can we use this for? Of course, we can use it to figure out prognosis. We can figure out ways of targeting MyCAF heavy tumor types. Some of those happens to be related to KRAS signaling. And in order to explore this biology a little bit more, we have recently went into publicly available datasets from KRAS-treated mice. And what you basically see here is super interesting. We are looking at pancreas cancer model, and you can see compared to healthy mice, we do see these MyCAF collagen recurring in the tumor-bearing mice. But when mice are treated with KRAS inhibitors, they completely disappear again. So we are looking at KRAS inhibitors that targets tumor cell with a clear bystander effect on the MyCAF burden, which is in itself an interesting biology, but possibly we can also use some of these serum biomarkers to track what will go on in all the clinical trials that are currently enrolling patients. And then finally, I've shown you how we can now already use some of our fibrosis biomarkers. If I have 1.5 minutes left, I will also show you that we have inflammatory tools that we can use to track immune cell engagement here. One of the tools we use a lot, in particular in immuno-oncology, is VICM. It's related to M1 macrophage activity. And what we see there is at baseline, when we measure VICM in circulation, if patients have high levels, they do better on an anti-PD-1 than they do on a tyrosine kinase inhibitor, for example, and indicating predictive potential. Within arm, on immunotherapy, high vacuum levels is better than low levels. So if you already have an inflamed microenvironment, you are better off on an immunotherapy in cancer. And of course, we can also track changes on treatment with this biomarker, as we do already from phase 1. And the other biomarker is C4G. This is basically a collagen fragment that is cleaved by granzyme B. So when CD8 T cells are active, they secrete granzyme B, integrate type 4 collagen. We can measure that in circulation. Initially, we saw proof of concept data indicating that if patients have higher levels of this biomarker baseline, they do better than if they have low levels. We are more using this now to track T cell engagement and track efficacy of immunotherapies over time. An example from CTC last year here is from cholangio, where we have patients treated with Nivo, EP, and radiotherapy divided into whether they were responding or not to treatment. The take-home message here is that all responders actually had an increase in C4G, or put differently, if C4G increased, indicating T cell engagement, you had a 45% response rate in your cohort, compared to if C4G did not increase, that was 0% that were actually benefiting from treatment. So we can use this as a surrogate of T cell engagement in immunotherapy trials. So this is a summary. I've presented you a snapshot of some of these collagen biomarkers that we use to assess CAF activity, immune cell engagement, that can give us an insight into tumor microenvironment alterations. It can support patient stratification, predict them, response to treatment, and importantly look at if and how we are modulating the fibroblast component. I want to acknowledge all my good colleagues and collaborators at Nordic Bioscience, but also all our external collaborators throughout the world, I would say, and also the patients, of course, to contributing with important serum and plasma samples for us to do the actual research here. So, yeah, with that, thank you very much.
Thank you, Nicholas. Impressive. And a lot of questions for that. Let me start with one question from you from the chat here. ProC3, is it because of drug resistance, or is there less penetration, or how come it's so prognostic and predictive?
Yeah, so there's definitely the drug barrier component to this, where the more ProC3 you have, the more buildup of matrix you will have, and that is basically a penetration barrier associated to that has been shown by many groups, that the more dense and overexpressed collagen you have, the harder it is for particular larger molecules to penetrate and reach the tumor nests. And we see this with antibodies, we see this with ADCs, we see this with other larger molecules. It is really those with low ProC3 that are benefiting more than those with high ProC3.
Thank you. I see Linda is really fast in answering questions in the chat, but I thought that was a super good question from Adriana and Linda. Can we do that online? Because I thought that was we do a lot of IPF at Nordic, and ProC3 is super prognostic as well. So question from Adriana, do you think the copper depletion could be an option for treatment of pulmonary fibrosis? I think that's a good question.
Yeah, I mean, well, I'm going to say that's okay. I'm not a pulmonologist, but as I recall, I don't think there's any effective treatment for pulmonary fibrosis. So I think that a trial of copper depletion in that setting to reduce fibrosis, I think would certainly worthwhile exploring.
Thank you.
I say yes.
There are many questions in there, and we unfortunately are running out of time because we have had 3 speakers that are super energetic, and you can see your energy is really coming out. So thank you everybody for dialing in. We will share all the questions with the speakers, and you'll hear from us getting back. Remember, these symposium are just to keep us warm until we have the real conference in Copenhagen, Denmark. Hoping to see many of you and submitting abstracts. And thank you everybody for doing extracellular matrix. It's super important that we tackle those problems for patients. Thank you, and have a good rest of the day and evening.
Thank you. It's been great to hear what everybody is doing and to see all the progress you guys have made in this field. You've created it. So
thank you. Thank you for joining in.